Welcome to Concept Biology. We are going to be discussing Chapter 6, Reproduction at the Cellular Level. So let's get into it. In this chapter, we are going to describe the prokaryotic and eukaryotic genome. As well as kind of distinguish between the differences between chromosomes, genes, and traits. Chromosomes, genes, and traits. And that's the genes, traits, stuff like that. That's going to play an important role coming up in inheritance, in genetics, things like that. How do cells reproduce? Sea urchins, for example, start as a single-celled organism that will then divide into two, that will divide into more and four, and so on and so forth until they become a whole organism. And that's a pretty similar process that human cell reproduction takes as well. So how did we become who we are? If we started off as a single cell, how did we become these full multicellular organisms? We all started as a single cell. That single cell then was fertilized and underwent a lot of division, a whole bunch of division, okay? We use that cell production for growth and development. If you remember from some of our earlier chapters, growth and development is one of the main themes of biology. Most all living things must be able to grow and develop. How do we do that growth and development? We do it through a couple of processes. One is sexual reproduction, the other is asexual reproduction. The differences between these are asexual reproduction does not require a partner. Okay. In the next kind of chapter, we're going to be talking about the differences between those two, and then the cycle that cells take to kind of grow, reproduce, and make more of themselves. But before we get into that, we need to discuss what is a genome. A genome is the cell's complete amount of DNA. So all of the DNA that a cell will have in its nucleus is called the genome. That DNA is made up of those nucleic acids, a ribose sugar, and a phosphate. Okay. In prokaryotes, DNA is a single double-stranded DNA molecule, and it's usually going to be in like a loop or a circle. But in eukaryotes, we have several. That's not how you spell several. Several double-stranded DNA molecules. And they are usually linear. So they're not kind of in those loops or circles. They're pretty just in straight lines. Different species will have a different number of these double-stranded DNA molecules. Okay. Human somatic cells, our body cells, will have 46 chromosomes. Can't spell. But our gametes, our sex cells, will have 23 chromosomes. You may be wondering, why do our sex cells have 23 chromosomes? That leads us to our discussion about homologous chromosomes and sexual reproduction. Okay? So, if our sex cells have 23 chromosomes, for females, that sex cell is the egg, and for males, that sex cell is the sperm. Okay? The combination of the egg and the sperm will get us to a total of 46 chromosomes. Okay? So a homologous chromosome is just a pair of chromosomes. One from one parent and one from the other parent. Okay? So a homologous chromosome is going to be a chromosome that has the same length, same centromere position, same everything. You just got one from one parent and one from the other. Central. Okay, so they are the same chromosome, just from a different parent. This takes place in all of our somatic body cells, that 46 chromosomes, except for our sex chromosomes. Our sex chromosomes are the only ones that will be different homologous pairs. Males will have an X and a Y, is a biological male, and an X is a biological female. Okay. The way that we show the differences in these chromosomes, the way we pair up our chromosomes, is via a image called a karyotype. And you'll note in the karyotype, you have all of these chromosomes, 1, 2, 3, 4, and you have doubles of all of them, except for this X chromosome. You have 2 X chromosomes, but you don't have a Y chromosome. So this person is genetically, biologically female. But all of the other chromosomes are the same. Okay? You have 23 total, 23 pairs, with 46 total. Okay? So now that we've kind of talked about homologous chromosomes, the genome, things like that, now we're going to talk about the cell cycle. The cell cycle is a cell's way of duplicating itself, duplicating everything in itself. To make another copy of the same cell. Okay, so in this section, we are going to describe the 3 stages of interphase. We're going to talk about how chromosomes react during mitosis and how the cytoplasmic contents divide during cytokinesis. We're going to talk about the quiet, almost imperceptible G0 phase. And then we're going to talk about the 3 internal control checkpoints that make sure that our cells are duplicating properly and not creating an excess or mismanaging their duplications. Okay. So this is an overview of the cell cycle. I want you guys to note that most of the cell cycle, so all of here, and all of here, are all interphase. All of that is interphase. And just this tiny little bit right in here is the actual mitotic phase. So most of the time, a cell is hanging out in interphase. Okay. Interphase has 3 parts that we talk about: the G1 phase, the S phase, and the G2 phase. G1 is just growth. S phase is DNA synthesis. And then G2 is another growth phase. Okay. DNA synthesis, that's basically where all of the DNA is going to copy itself. Okay. Mitosis is where the actual chromosomes are going to be pulled apart. And mitosis has 5 stages. There's prophase, prometaphase, metaphase, anaphase, and telophase. And then the last unofficial step of mitosis is cytokinesis, and this is where the cell actually breaks apart. And we're going to talk about all of these different phases in a bit more detail. So the first phase of mitosis, prophase. In prophase, the things that happen, the nuclear envelope breaks down. The vesicles, the Golgi apparatus, the endoplasmic reticulum, all of those organelles start to kind of disperse. And the nucleolus disappears. Okay. In prometaphase, second phase, prometaphase, we kind of just continue the things in prophase. The rest of the very bits and ends of the nuclear envelope fully disappear. The mitotic spindle starts to develop. And the chromosomes become a lot more condensed. In prophase, your chromosomes are still kind of spaghetti. Okay. Then in the last kind of bit of prometaphase, the sister chromatids will attach to the spindle microtubules via a kinetochore. So sister to kinetochore. Okay. And then in metaphase, the chromosomes are going to line up in the middle of the cell at what we call the metaphase plate or the equatorial plate. Okay. Then the fourth phase, during anaphase, the sister chromatids are pulled apart by those kinetochores, by the centromeres that were attached to the kinetochores. Okay. Apart and then pulled towards the centrosome towards. Okay. And then the fifth phase, telophase, all of the prophase and prometaphase stuff gets reversed. So the nuclear envelope comes back, the organelles come back, the nucleolus comes back, the chromosomes go back into being spaghetti, they're hanging out. Okay? The mitotic spindle is broken down, and the nuclear envelope reforms. And then cytokinesis, that very, very last official, unofficial phase, is where the cells will start to actually physically break apart. In animal cells, that looks a little bit different. Animal cells get what we call a cleavage furrow, and plant cells get a cell plate that will eventually turn into a cell wall. This cleavage furrow turns into a pinch. Pinch, pinch. Okay. Just a couple of differences to be aware of. In an animal cell, when we're trying to separate the cells, what we call cleavage furrow forms. It's kind of like the pinching of those two cells. And then in a plant cell, a cell plate forms in between the two nucleuses that will eventually turn into the cell wall in between those two cells. The G0 phase. G0 phase is just when a cell is not actively trying to divide or do anything. This is kind of a holding phase. Okay. And different cells will take different amounts of time to divide. It's super variable between the cells of an organism. So epithelial cells, these are things like your skin cells, linings of membranes, etc. Those will take about 2 to 5 days. Every 2 to 5 days, they will divide. Neurons, cardiac muscles, they don't do a lot of division after you're fully grown. And the cell cycle takes around 24 hours. Okay. That cell cycle is heavily, heavily regulated. One of the ways that we regulate it is with the internal checkpoints. So there are some checkpoints in between G1 phase going into the S phase. So if the cell is not growing properly, if there's something messed up with how it is functioning, it will stop the cell and send it out for apoptosis, which is a programmed cell death. Okay. If it makes it past that G1 checkpoint, it will synthesize all of that DNA and go into the G2 checkpoint. If at that G2 checkpoint, the cell finds that there is something wrong or there's something that is not made correctly either in the DNA or in the cell growth itself, it will again get tossed out for apoptosis. If not, it will go on to mitosis. And there is one more checkpoint at this mitotic phase. If the cell does not properly separate the chromosomes, the cell will then get tossed again for apoptosis. What happens if some of these checkpoints don't work? Because as much as this cell reproduction is a very highly regulated, a very consistent machine, it is not perfect. So what happens when some of those internal checkpoints do not play in properly? We get things like cancer. So in this section, we're going to explain how cancer is caused by uncontrolled cell division. We're going to understand how proto-oncogenes become oncogenes. We're going to describe tumor suppressor functions and how they work to stop certain events. And then we are going to explain how mutant tumor suppressors can cause cancer. Okay. So what exactly is cancer? Cancer is just your own cells that have divided uncontrollably. Okay. So either the checkpoints didn't work, you have genes that are pushing, that got mutated and are pushing mutated and pushing this process forward, etc. There's a whole bunch of things that could cause cancer. Okay. Those mutations or those errors can then be passed on from one cell to the next, to the next, to the next, which just makes things worse. Okay. So the changes that a cell could make could be very minor. It could just be a single little nucleotide that got moved over, that got copied wrong or incorrectly, something like that, something small that could eventually change how more cells make their proteins and then how they function later on down the line. And so from that uncontrolled growth, those mutations in that DNA change the proteins, change the cell, and can result in a tumor. Proto-oncogenes are genes that code for the positive cell cycle regulators. So these are helpful. We have a lot of proto-oncogenes that control the cell cycle. Okay? Those proto-oncogenes are normal until they get mutated. It is possible for them to, if the DNA synthesis happens incorrectly or a nucleotide is replaced where it shouldn't be, that proto-oncogene can be mutated and become an oncogene. And that is a gene that will cause a cell to become cancerous. Okay? If a cell can't reproduce, that mutation does not move on to other cells, and the problem pretty much stops there. But if that mutation does cause a change in a positive regulator, it can force the cell to go crazy and just multiply like crazy. Multiply like crazy. Without stopping, without checking, without any kinds of the checks and balances that are usually in place, those mutated oncogenes will force the cell to just divide. That's the only thing that it'll tell you to do. We, however, have tumor suppressor genes, and these genes code for the negative regulator proteins, the type of proteins that can prevent a cell from undergoing uncontrolled cell division. Okay? Sometimes it is the tumor suppressor gene that gets mutated. And if that tumor suppressor gene gets mutated, the cell will not be able to stop the cell cycle if there's a problem. Okay. So the protein P53 is one that usually monitors DNA. And once, if that P53 is functioning normally, this is a proto-oncogene, by the way. When it's functioning normally, it will stop the cell cycle, either repair the DNA and restart the cell cycle, or send it off to apoptosis so that that cell dies. If that P53 is damaged or mutated, the cell cycle will just continue. It'll just go and go and go and go and go, and then the cells can very quickly become cancerous. So how do we treat cancer? We will either cut it out. It's a whole bunch of cells. We can see which cells are normal, which cells are abnormal. We'll cut it out. That's surgery. We can burn it. That's cauterization. Or we can do radiation, which is using lasers to zap it. Or we can do chemotherapy, which is using chemicals to kill it. These two are slightly more dangerous because they are not super discriminative, meaning that they will also affect your normal cells. Okay? These can be curative, but aren't always. If you miss even one cell, the thing can grow back, and that's not exactly what we want. Prokaryotic cells, they also do cell division. So we're going to talk about a little bit about binary fission and explain how the FTSZ and tubulin proteins are examples of homology. Homology just means similar structure function in a different kind of existence. Okay. So with the process of binary fission, this is a form of asexual reproduction. And in that asexual reproduction, those daughter cells are genetically identical to the parent cells. Okay. Mitosis is not necessary because there is no nucleus or multiple chromosomes. It's just one singular circular chromosome that can get reproduced and divided quickly. So that bacterial reproduction happens very, very fast. And this is kind of that process of binary fission. You guys can read over this and it'll explain that pretty well. We don't really talk about binary fission too much because it's prokaryotic in nature. You just need to know that bacteria do it. They do it really fast, and it's pretty similar. The DNA gets duplicated. The proteins kind of migrate towards the middle. That cleavage furrow forms. You form a septum, and the cell breaks apart. And that's pretty much it for cell growth.